Today I'll be speaking about some work on guard cell specific alternative splicing during the drought response in Arabidopsis thaliana. So for the non-plant scientists in the room, one of the major goals of plant science is to address global food security, particularly in light of climate change. So climate change poses threats to our food supply, and one of the biggest is drought stress. So the incidence of drought stress is projected to more than double by the end of this century, largely affecting areas with wetter climates. That includes us. So guard cells are the plant's primary cell type that it uses in the drought response. So guard cells exist in pairs on the surface of the leaf, and they open and close dynamically to allow for gas exchange and transpiration. During drought stress, production of the phytohormone abscisic acid triggers a signaling cascade, and this results in ion channel activation and a net anion efflux from the guard cells, decreasing trigger pressure and causing them to collapse over the stomatopore, shutting off both water loss and gas exchange. This also leads to widespread transcriptional reprogramming, both at the expression level and at the splicing level. So previous work in our lab by Anna Van Weren aimed to profile the guard cell response to physiologically relevant progressive drought stress. So over about a 2-week time course, Anna samples plants at 60, 40, and 20% soil water content, corresponding to decreasing water availability to the plant. And the system she used was called INTACT. So INTACT is a nuclear membrane localized biotin tag expressed under a cell type specific promoter. And through this system, Anna was able to select out guard cell nuclei and compare them to a bulk leaf control under a constitutive expression promoter. So the first thing that I kind of did when I joined the lab was take a genome-wide survey of alternative splicing between the guard cells and the bulk leaf tissue, and also across the drought time course. So if you can see, the D's correspond to drought, the W's to water, and the numbers to the water availability or soil water content. So we've identified some drought responsive splicing events. For instance, retention of this intron is increased in guard cells during drought stress. And we also identified guard cell specific events. So retention of this intron is actually decreased in guard cells where the splicing is more efficient. And this data actually is freely accessible, and you can investigate it yourself using the BARS EFP-seq browser. Following the genome-wide analysis, we wanted to understand what actually happens to alternative transcripts in the cell. So as I'm sure you know, alternative splicing can give rise to alternative protein isoforms. But in plants, it's actually more common that splicing regulates the mRNA itself. So nonsense mediated decay is a mRNA quality control process that regulates transcripts with premature stop codons or other deleterious effects to the coding sequence. And we also have nuclear retention, which is itself an end process, or can lead to post-transcriptional splicing and further targeting to one of the other two pathways. So we followed transcripts through each of these 3 pathways using subcellular fractionation and various mutant lines. And we found that, unsurprisingly, the majority of transcripts don't seem to give rise to an alternative protein product. And most alternative splicing is instead regulating transcript levels at the mRNA level. So we asked, why do some genes evolve to be regulated by splicing instead of by expression? When we compare differentially expressed and differentially spliced populations in both cell types, they increase with increasing drought severity. But when we looked at lists of the genes themselves, we actually found that the overlap was quite minimal. So given the size of the Arabidopsis thaliana genome, this overlap of 130 is about what we expect by random chance. And so it appears that the 2 processes regulate distinct sets of genes. To understand what these genes do, we performed gene ontology analysis, and we actually found a great deal of overlap in some of the key functional processes that these genes regulate, including processes associated with the response to abiotic and osmotic stimuli, and also transcription and translational regulation. So while differential splicing and expression act on different lists of genes, it seems that these genes have similar functions as well. And to understand why one regulatory process may be preferred over another, we took inspiration from animal studies that have postulated that gene pleiotropy may play a role in the preferred regulatory pathway, where genes with high pleiotropy are ill-suited to regulation by expression, and instead may be regulated at the splicing level. So we use tissue specificity as a proxy of pleiotropy, reasoning that genes with a broader expression pattern may have more functions in the organism. And indeed, we were able to show that differentially spliced genes have a lower tissue specificity index corresponding to broader expression and potentially corresponding to a greater degree of pleiotropy in the plant. So this is kind of one avenue of reasoning for why some genes have evolved to be regulated at the splicing level. Another kind of way that we've been thinking about this comes from a separate project, which is looking at NMD eliciting transcript features. So NMD is nonsense mediated decay, and it's one of the key outcomes of plant alternative splicing. And the way that plant scientists tend to study NMD is using NMD deficient lines. So UPF1 is an RNA helicase that's a core component of the NMD pathway. And when it's knocked out, we lose NMD in the plant. And so what we see is this increase in transcript expression for NMD sensitive isoforms. So using two separate data sets from two different experiments, we analyzed their RNA-seq data to generate an NMD sensitivity score. And then for these transcripts, we extracted features describing their sequence and structure from the Arabidopsis thaliana reference transcript database version 3, which contains information on about 170,000 transcript isoforms identified using long-read RNA sequencing. So from the transcript features and the NMD sensitivity score, we developed a random forest regression model to try to understand what are the key transcript features that drive NMD sensitivity. So the first thing we noticed about the results from both models is that they appear quite similar, and that's reassuring because of the different genotypes and also the different developmental stages. But what we're looking at here is the percent of variance explained by groups of features that were inputted into the model. So in this orange color, we have features associated with the CDS and the 3' UTR. So this is where we find things including premature stop codons, degree of CDS truncation, and also splicing variants in the 3' UTR, all of which are known NMD listeners. And so we expected these features to explain a great deal of variance, and that is in fact what we saw. But what we didn't expect was this really critical role of expression level. So it seems that the wild type expression is a strong predictor of NMD sensitivity. And so to understand the direction in which that effect happens, we basically took out the bottom 20% of NMD targets and the top 20%, so the most insensitive and the most sensitive transcripts. And what we find is that NMD sensitive transcripts tend to have much lower expression, both in terms of their TPM, so transcripts per million, as well as in their isoform fraction, so whether they constitute a major or minor isoform. So all of this to say, gene expression might be another variable that plays into the choice between the preferred regulatory pathway, because NMD is a key output of alternative splicing. If a gene is a strong NMD target, then perhaps that's why it's evolved to be regulated at the splicing level, as opposed to the expression level. And then just to wrap up, I wanted to bring it back to drought. So these are two of our guard cell specific differentially spliced genes that we identified. And as you can see, there's an increase in the separation between the isoform ratios at 20% soil water content. So these are responsive to severe drought. We obtained two independent knockout lines for each of these genes and assessed their drought susceptibility by measuring the relative water content under severe drought stress. And so the decrease in water content indicates that the plant is really struggling to maintain its physiology under severe drought, pointing to an increase in drought susceptibility. We also saw a impaired response to rewatering. So after severe drought, when we rewatered these knockout plants, they didn't respond as well as the wild type, which is 0. So I think this highlights two really cool points. The first of which is that these splice variants were only identified in the guard cell samples, which we think really highlights the utility of these cell type specific approaches in identifying key regulators of stress responsive pathways. Second really cool thing is that when we track these transcripts through the cell, we actually found that they were regulated by two distinct pathways. So safeguard one appears to produce a second protein isoform. So work in our lab is ongoing to confirm the presence of this protein, while XTH31 is NMD sensitive. So the two different pathways both seem to regulate key drought responsive transcripts in the cell. Yeah, so just to conclude, drought induces these guard cell specific changes that regulate the transcriptome more so than the proteome. Alternative splicing may have evolved to regulate certain subclasses of genes based on their pleiotropy or expression level. Guard cell specific alternative splicing regulates really key functional genes in the drought response. And some future work in our lab that we're really excited about, perturbing the expression levels of NMD sensitive transcripts and assaying for their resulting NMD sensitivity, we hope will shed some light on the true role of expression in the NMD pathway.